the way the talk is structured is exactly the way you saw it when you signed up. Basically, brief overview over our technology, then some application examples, and yeah, if you have any questions, of course, we're here to answer them. So first of all, the core of our epitope mapping services is our microfluidic chip platform, and currently we have two different chips. They differ in terms of in the number of features. What is shown here is our smaller and the original chip. It has 4,000 features and a total volume of only 10 microliters. So the way the chip is constructed, I'll go into details in a little bit, but just so that you have an overall impression here already. It's a silica-based chip where the channels and reaction chambers are etched in, and the whole chip is covered with glass, except for one inlet and one outlet. It's pretty much a closed system. So here on the right, you see the electron microscopic picture of the liquid channels here and the reaction chambers. So the liquid gets pushed through at a uniform flow rate so that the reaction chambers are all flooded. As I mentioned, this is the smaller chip. We have also a larger one that has up to 30,000 features, but typically for the average epitope mapping project, the 4,000 different peptides are plenty. So there are basically three things that allows us maximum flexibility with this platform. One point is for sure the chemistry, the way we're doing the photochemistry. The second point is the fact that we are using digital photolithography. And then the third part, of course, that we're using microfluidics. And I will touch on each of these topics now on the following briefly. So the chemistry, the way we are doing the synthesis is using photochemistry. So basically by using UV light that we hit each individual reaction chamber with. And typically the way it's done is you use specialty monomers that are photolabile. And basically when the UV light hits the building block, the amino acid, the protection group dissociates and synthesis occurs. So that limits you in terms of the amino acid building blocks that you can use. And of course you're looking at specialty amino acids, so it's pretty expensive. We, on the other hand, we can use just the regular standard DMT chemistry and use basically any typical default or standard commonly used FMOG or BOC, TBOC chemistry reagents. And so the way we are starting the synthesis is using a photogenerated acid. So in our case, the acid which catalyzes the reaction is actually the photolabile part, and that's what's shown here. That's the dissociation of the precursor acid into the PGA, the photogenerated acid. And here, just another visual, what I just mentioned is that we are using standard TBOC protecting groups versus expensive and rather limiting other photolabile protecting groups. So no matter what type of special modification you have in your case, typically as long as it's somehow commercially available, we can use it in our synthesis. Or if you have it from your own lab, of course, you can send it to us. And the second part is the digital photolithography. What this means is basically that a computer is used to generate the mask that is then projected onto the chip and regulates where the UV light hits the chip and where not. So here on the right-hand side, you see the chip, and then these light rays are symbolizing where these columns, the lighter columns, are symbolizing where the UV light hits the reaction chamber in this particular synthesis step. And yeah, so this is regulated by a mask, which used to be an actual physical mask, but nowadays, of course, computer combined with a projector system are being used for that. So this allows us to really do on-demand synthesis and completely customize it. So we don't have any pre-made chips on the shelf. Every single array that we are running is a custom-made array. And here just a quick animation how this works. Basically here you have the chip with a silica substrate with the edge channels and reaction chambers in it. And this is covered with glass. And you have one inlet here and then the outlet here. And for each synthesis step, of course, we flood the entire system with the building block that we want to attach in this step. And then the mask gets projected on the chip, and UV light hits only in those reaction chambers that we intend to synthesize. Of course, that is that's just one cycle, so this whole procedure repeats for every single amino acid. And so typically a synthesis time is anywhere between 5 to 10 days, depending on the length of the peptides used, and as well as the sequence variation that is in the chip layout. Then the third part is the fact that we're using microfluidics. It allows us to run multiple washing steps or multiple binding steps at different protein concentrations. We can vary the temperature, and the nice thing is that we can change all these parameters while keeping the other parameters constant. So the data quality is rather good. Regarding the multiple washing steps, I will talk about that a little bit more when it comes to increasing doing multiple runs at different protein concentrations. It's typically the way that we, of course, start out with a lower protein concentration or sample concentration, and then slowly increase it. Because once your sample is bound to the chip, it's typically very difficult to strip from the peptide probes. Just to give you a visual impression of the difference between microfluidic chips and spotted arrays, typically peptides are being synthesized initially in a pool and then spotted on glass slides. So these are just some comparison of some raw data. This is here a typical spotted array. You see the problem that you have with the donut-shaped, really highly increased intensity in the outer edge of the drop, and then the intensity is significantly lower in the middle of the spot. And if you compare this to the standard chip of ours, it's you see that the signal intensity is much more uniform in this case. These drops here to the left and right are actually just are not even taken into account. We just integrate the middle part here. So these drops here symbolize the in and outlet of each reaction chamber here. So to summarize the comparison, the spot uniformity is clearly superior for a microfluidic chip. You have also pretty good sensitivity because of an enhanced mass transfer, because the sample gets constantly circulated. And you don't have any dye oxidation, so you don't have any issues that you're scanning the chip and then a week later it would give you completely different results. So because it's pretty much a closed system, you can actually put it really in the drawer and take it out 3 months later and either reuse it or, of course, scan it again, and you would get the same intensity readings. So this is a brief overview over the technology, and so in terms of synthesis, we're not just limited to peptides. Of course, that's what this webinar is about. So in terms of peptide arrays, we clearly the most we are running most of the experiments for epitope mapping purposes, or also in general protein-protein interaction arrays. Another option would be, another application are kinase substrate arrays, where we basically synthesize all potential kinase substrates on the array and determine the substrate specificity of purified kinases. And then something that we've gone into recently are phosphopeptide arrays. So the technology is the same for all of these applications, and in the past we have done the vast majority of our experiments have been in the DNA and RNA field, particularly microRNA arrays. So when you go to our website and look at the customer publications, because we start out in the microRNA field, that's why you simply find the vast majority of publications in this field, and only a few so far in the peptide field. So if you look at epitope mapping approaches using our array technology, there are, of course, mainly three that come to mind. One would be peptide scanning or tiling arrays. Basically, you're taking the target protein sequence and tile it in whichever amino acid resolution you would like on the chip. By default, we would use, because we have plenty of space on the chip, we typically use single amino acid resolution. Then another thing you might be interested in are truncation studies, basically from the N or C terminal truncation, from the N or C terminal end. Or then once you've identified the epitope, or you're getting closer, then of course also alanine scans or any other type of mutational scans. And I will show you now some data for a case study that we did. Before that, just the typical parameters. By default, we are using 8 mers in terms of probe length, but we can synthesize longer peptides up to 12 mers. And the reason why I put here on the slide variable region is simply that we don't want to synthesize more than 12 amino acid residues in a row using photochemistry. Beyond that, the yield simply drops dramatically. If, on the other hand, you have some kind of constant regions for all peptides, let's say you have a 5 amino acid residue length of common sequence for all of the peptides, then we can certainly add that either at the C or N terminal using standard chemistry. Then, yeah, typically we tile in single amino acid resolution. And what's shown here on the right is just a chip surface with a linker attached. The linker is actually kind of misleading here because the length is actually corresponds to roughly 35 amino acids in length. So it's much more, it's not really proportional, this graphic here. The linker is actually much longer. And then on top of the linker, we have then attached to the C terminus the actual peptide. And so the N terminus is the free end of each peptide. When we run the arrays, typically what we're using are 100 microliter of sample volume. That's for the smaller chip. The larger chip would require a little bit more. Then in terms of sample concentration, the good measure is typically 1 microgram per milliliter. So really not a lot. If you are having serum samples, we would typically we would dilute the serum 1 to 1,000 and see what type of data we're getting with that. And if this is too weak, doesn't show enough binding, then we would go ahead and run on the same chip a dilution of 1 to 100. And if this is still too weak, then we would dilute it 1 to 10. Typically, the best results so far we are getting with 1 to 100 dilution with serum. Then, depending on the chip that you're using, or depending on the project, depending on how many unique sequences, peptide sequences you need, you would go either with a smaller chip, which can hold up to 3,918 unique peptide sequences, or with a larger one, in case you're screening a lot of target proteins at once, which can hold up to 30,000 individual peptides. The largest chip can be also run as 6 individual, almost 5K chips. The way this works is basically we fill up the chip with the sequences and the tiling that the customer designs and the number of repeats they ask us for, but then we just fill up the chip with additional repeats until until it's full. When it comes to the detection step, we are using Cy3 or Cy5 labeled secondary antibodies. Of course, you could send us also your directly labeled antibody. Or in other applications, of course, we're using enzymatic stains, like for the kinase arrays. But in general, we would prefer to have a Cy5 labeled secondary antibody or primary labeled antibody. Cy3 works as well, though. It's just that the background for Cy5 is considerably lower. And as mentioned, a brief case study. So these are the following experiments were all done to determine, basically to validate the platform. And we looked at the anti-HA binding epitope, which is the influenza hemagglutinin epitope. And so we used 2 chips to look at this. And this is just a glimpse of one of the chips here that's shown here. So, and we put on the chip the known epitope and then a couple of variations, a flag epitope and all its variant sequences. And we had at least 3 replicates. When you're looking at the left side of this picture, this is basically a chip scan after pre-staining, basically like just the background control. You see it's basically black, no binding whatsoever. And then here on the right hand side, this is the experiment or the scan after we used the anti-HA antibody, and we used it actually at 1 microgram per milliliter. We did multiple runs. Starting with 0.1 microgram all the way up to 1 microgram per milliliter, and I show you some data from that in a little bit. And then detection in this case was Cy5, that's why it's red. And the result of this was really that the known epitope was clearly confirmed, and the 3 replicates had a variation of roughly 15%. So fairly good agreement. And then, in addition to just looking at the different variant sequences and unrelated peptide sequences, we also did truncation scans, one from the N-terminus, then second from the C-terminus, and then another one where we truncated simultaneously from both sides. And so the chip 1 and 2 are referring just to different probe content on the chips, but these were pretty much the only ones that actually were only peptides that were detected. So none of the unspecific, unrelated probe content that we had put on the chip was detected in a significant signal intensity. So looking at the N-terminal truncation, it becomes clear here that the residues shown in here on the right side, the first 4 are basically not are critical. If these are missing, you don't have any binding, and here you have really high binding intensity once you reach the core domain here of the sequence. Then the C-terminus, on the other hand, just 1 residue is critical. And let me show you the titration experiments for this. This is basically shown the N-terminal truncation scan. The peptides are listed here. So these are all the truncations. And then we did, each data point represents an assay at a particular increasing antibody concentration. So this is how you get pretty nice titration curves like this. And it basically reflects exactly what was shown here on the left side with the internal region, that you have basically no binding whatsoever and then suddenly an increase in binding. It's exactly shown here with the titration experiment as well. And then when you go one step further, you can of course then focus on alanine scanning or any other type of mutation that you would like to do. And these are the results of the alanine scanning. So it's clear that these three residues here are essential. So that would be the D, D, Y here marked in red. And of course, what we looked at then is we compared these data with the published Epitope data, and that were done in solution, and they were in perfect agreement. And just one more brief case study. This was basically using real-world samples from, these were actually patient samples. So this was an epitope mapping of SMBB, and the samples were was an antibody sample isolated from patient sera, and the published epitope is noted below here. It's PPGM-RPP, and if you look at the intensity data, that's exactly what we found here as well with our array. So the epitope is represented 3 times in the amino acid sequence, in the protein sequence. That's why it shows up here 3 times. Beyond the titration experiments that I just mentioned with the truncation studies, where you basically use one chip and run it multiple times at increasing protein concentration, you can also do something else and run it at a fixed protein concentration or antibody concentration. Basically, with just a single constant sample concentration, you can get the same data. And the way this works is that we vary, in this case, the peptide density or concentration on the chip. So what is shown here on the left are basically individual wells on the chip. And as you can see here, the protein concentration is, the peptide concentration is increasing towards the back. And basically the way we are doing this is using photochemistry. Is we are regulating the amount of light, and also we're using other measures to control this, but basically we are able to synthesize the same peptide multiple times on the chip at a different density. So this then allows for binding affinity measurements using just one chip, one assay at a fixed sample concentration. And here on the right-hand side is shown exactly this. It was the same case study as mentioned before. It was an anti-HA antibody, and the epitope was known. And here we've got a really nice binding curve. So this is almost perfect here. Typically, we use 8 different density spots. That's pretty much the limit. You want to have at least 6 different peptide densities usually. And just to finishing up, just to show you some real-world chip scans from a project that we did recently for a customer. So this was a project where they had particular target protein, and they had a whole wide array of different antibodies, and they want to know which ones are actually specific binders and good binders. So the purpose was to find the optimum antibody. And what's shown here right now is just a negative control, basically a blank chip, after probably washing it with some let's say some serum or so that didn't include the antibody. Then this is the data for weak binder. So the color code is basically dark blue is the background. It's an intensity value of 0, and bright red or orange would be the highest intensity of roughly 65,000. So what you're seeing here are just light blue spots, and you see there's an increase here and then it goes down again. So this might be an epitope, but it's the binding is just very weak. So then another case is this one, which is just shows a lot of binding and looks just too much and rather unspecific. And then the ideal case is an antibody like this, where you have only very select, highly specific binding to the epitope. So these just as a few examples. Just briefly about us. We are located here in Houston. There's the headquarters. We have also offices in China and representations in other countries, but all the experiments and all the samples end up here in Houston in our lab. We are doing custom microarrays since 2005, and we processed way more than 12,000 samples by now. Most of these samples are, as mentioned before, in the RNA field, microRNA especially. But we started offering peptide array services a few years ago, and we are now focusing on this as well. The technology for all our microarrays is the same as outlined at the beginning of the webinar. It's what we call the microParaflo microfluidic technology. We have plenty of customer publications, as mentioned, mostly in the microRNA field so far, but that is changing hopefully soon. We have customers all over the world, and depending on where you are, you can contact either your local representation or send the samples directly to us. Especially our customers in Europe send us directly their samples. The main reason for people to contact local distributors would be shipping-related issues. Yeah, we receive samples from 40 different countries directly here, so that's really not a problem any longer.